The embryology of the female genital tract forms the foundation for understanding both normal anatomy and many congenital disorders encountered in clinical practice. Unlike the adult reproductive system, which appears as a single integrated unit, the female genital tract develops through a series of coordinated embryological events involving the gonads, paired duct systems, and external genital primordia. A clear understanding of these developmental processes makes it much easier to understand congenital anomalies such as Müllerian agenesis, uniconnuate uterus, biconnuate uterus, septate uterus, and disorders of sexual differentiation. In this module, we will study the development of the gonads, the Müllerian and Wolffian ducts, and the process of sexual differentiation that ultimately gives rise to the female reproductive system. Let us begin with the earliest stage of development, when the reproductive system is still in its indifferent stage. Development of the reproductive system begins during the 4th to 6th weeks of embryonic life. At this stage, male and female embryos are morphologically indistinguishable with respect to their reproductive organs. Both possess an indifferent gonad, a pair of Müllerian paramesonephric ducts, a pair of Wolffian mesonephric ducts, and a common set of external genital primordia. In other words, every embryo initially possesses the developmental potential to form either male or female reproductive structures. Sexual differentiation has not yet occurred. The direction of development depends on the genetic and hormonal signals that act during the following weeks. To understand how the female reproductive system develops, we must first examine the earliest event in this process, the formation of the indifferent gonad. The development of the gonads can be understood in 3 simple steps. First, the embryo forms a temporary structure called the genital or gonadal ridge. This appears during the 5th week as a thickening on the medial side of the mesonephros. At this stage, it is simply an indifferent gonad. It has the potential to become either an ovary or a testis. Second, this gonadal ridge must receive its future germ cells. An important point to remember is that primordial germ cells are not formed inside the gonad. Instead, they originate in the wall of the yolk sac. From there, they migrate through the hindgut and dorsal mesentery before finally reaching the gonadal ridge during the 5th to 6th weeks. Think of the gonadal ridge as an empty house. The house is built first. Only afterwards do the future occupants, the primordial germ cells, move in. Without these cells, normal gonadal development cannot occur. In rare cases, if some primordial germ cells fail to complete their journey, they may remain outside the gonads and later give rise to extragonadal germ cell tumors or teratomas. Once the gonadal ridge has been successfully populated by primordial germ cells, it is ready to differentiate into either an ovary or a testis. Once the gonadal ridge has been populated by primordial germ cells, the next question is simple. Will it become an ovary or a testis? The answer depends on the genetic signals received by the indifferent gonad. If the SRY gene is present and active, the gonad differentiates into a testis. But what happens when SRY is absent? The gonad does not simply remain unchanged. Instead, it actively begins developing into an ovary. This is an important concept because many students are taught that the female pathway is the default pathway. That statement is only partly true. It is true that, in the absence of male hormones, the embryo develops female internal and external genitalia. However, ovarian development itself is an active process. Genes such as WNT4 promote ovarian differentiation while suppressing the pathways that would otherwise lead to testicular development. As development continues, the cortex of the gonad becomes more prominent, while the medullary region gradually regresses. The primordial germ cells become surrounded by supporting cells, forming the primordial follicles. which constitute the basic functional units of the ovary. So remember this concept rather than memorizing isolated facts. No SRY does not mean nothing happens. It means the embryo actively follows the ovarian developmental pathway. Memory shortcut: SRY present leads to testis develops. SRY absent leads to WNT4 takes over, leads to ovary develops. Don't remember female is the default. Remember, female development is an active program in the absence of SRY. The next major event is the development of the Müllerian ducts, also known as the paramesonephric ducts. These ducts begin to develop around the 6th week. Initially, there are 2 Müllerian ducts, one on each side of the embryo. The easiest way to remember their development is to think of them as 2 railway tracks. For most of their course, the 2 tracks run separately, but as they descend into the pelvis, they gradually move towards each other and fuse in the midline. Now remember one simple rule: the part that remains separate forms paired organs. The part that fuses forms single midline organs. So, the unfused cranial portions remain separate and develop into the right and left uterine tubes. The fused caudal portions unite to form the uterovaginal primordium, which later develops into the uterus, cervix, and the upper part of the vagina. There is one final step. Although the ducts have fused, a partition, or septum, still separates them internally. This septum must gradually disappear through a process called septal resorption, creating a single uterine cavity. Instead of memorizing every derivative separately, remember just these 3 steps: formation, fusion, resorption. Almost every congenital Müllerian anomaly results from failure of one of these 3 developmental events, a concept we will explore in the next module. Memory shortcut: Müllerian ducts equals 3 F's formation, fusion, final septal resorption. That's all you need. If you remember these 3 steps, understanding congenital uterine anomalies later becomes remarkably easy. So far, we followed the development of the Müllerian ducts. But remember, the embryo also possesses another pair of ducts, the Wolffian ducts, also known as the mesonephric ducts. In the female embryo, these ducts do not disappear because they are unwanted. They disappear because they do not receive the signal needed to survive. The Wolffian ducts require testosterone for their maintenance. In the absence of testosterone, they gradually regress. This gives us a very easy way to remember both duct systems. No AMH, the Müllerian ducts survive. No testosterone, the Wolffian ducts regress. However, regression is rarely complete. Small portions of the Wolffian duct may persist as embryological remnants. The 3 important remnants to remember are epuphoron, paruphoron, Gartner duct. Among these, the Gartner duct is the most clinically important. If it persists, it may later form a Gartner duct cyst, which is typically found along the lateral wall of the vagina. An interesting clinical point is that Gartner duct cysts may sometimes be associated with renal or ureteric anomalies. This is not a coincidence. The urinary and reproductive systems develop side by side during embryonic life, so developmental abnormalities in one system may be accompanied by abnormalities in the other. So instead of memorizing 3 remnants separately, remember one concept. The Wolffian duct mostly disappears, but it occasionally leaves behind footprints. Memory shortcut: Wolffian duct equals testosterone keeps it alive. No testosterone equals regression. Regression is incomplete equals epuphoron, paruphoron, Gartner duct. At this stage, the embryo has everything it needs to develop into either a male or female. The final outcome depends on a few key signals. Let's understand them one by one. The first signal is the SRY gene. If the SRY gene is present and active, the indifferent gonad differentiates into a testis. Once the testis is formed, it begins producing 2 important substances. The Sertoli cells produce anti-Müllerian hormone, or AMH. As the name suggests, AMH causes the Müllerian ducts to regress. The Leydig cells produce testosterone. Testosterone maintains the Wolffian ducts, allowing them to develop into the male internal genital tract. A part of this testosterone is further converted into dihydrotestosterone, or DHT, which is responsible for the development of the male external genitalia. Now let's look at the female embryo. In the absence of SRY, the gonad develops into an ovary. Since there are no fetal Sertoli cells, AMH is not produced, so the Müllerian ducts persist. Since there are no fetal Leydig cells, testosterone is not produced, so the Wolffian ducts regress. And because DHT is absent, the common external genital primordia differentiate into the female external genitalia. Notice something important. The embryo does not begin with separate male and female reproductive systems. It begins with one common blueprint. Different genetic and hormonal signals simply guide that blueprint in different directions. Memory shortcut. Think of sexual differentiation as 3 simple questions. Is SRY present? If yes, a testis develops. Is AMH present? If yes, the Müllerian ducts regress. Is testosterone present? If yes, the Wolffian ducts survive. Is DHT present? If yes, the male external genitalia develop. No SRY leads to ovary. No AMH leads to Müllerian ducts persist. No testosterone leads to Wolffian ducts regress. No DHT leads to female external genitalia develop. Now let's bring the entire developmental sequence together. The female reproductive system does not develop from a single embryonic structure. Instead, it is assembled through a series of carefully coordinated developmental events. First, the gonadal ridge forms and is populated by primordial germ cells, allowing the indifferent gonad to develop. In the absence of the SRY gene, the gonad follows the ovarian pathway. At the same time, the Müllerian ducts persist and develop into the uterine tubes, uterus, cervix, and upper vagina. The Wolffian ducts, lacking testosterone, gradually regress, leaving behind only a few embryonic remnants. Finally, in the absence of DHT, the common external genital primordia differentiate into the female external genitalia. If you remember the sequence rather than isolated facts, the entire topic becomes much easier. Think of female genital embryology as a series of simple developmental decisions. Build the gonad. Populate it with germ cells. Differentiate it into an ovary. Preserve the Müllerian ducts. Regress the Wolffian ducts. Differentiate the external genitalia. Every congenital anomaly that you will study later can be traced back to one of these developmental steps. In the next module, we will see what happens when these processes fail, resulting in congenital anomalies of the female genital tract.